Hi students, this is A. Mandri Kumar Rajesh. In this video, I am going to teach you the next non-Mendelian inheritance cross called codominance. Let's begin the topic with the definition of codominance. The alleles do not show dominant or recessive relationship and are able to express themselves independently and equally when present together. That means the alleles can express themselves independently and equally when they are at heterozygous condition. This is called codominance of alleles or codominance. Then the event of codominance was reported in the plant called camellia. So the event codominance was reported in the plant camellia. Then I am going to figure out the cross. So the parental phenotype of the two individuals is red, white. Then the genotype of the dominant is capital R. For the recessive is capital W. Then the gametes produced by the dominant is capital R, here capital W. Fusion takes place, results in the development of F1 generation, and it shows the genotype capital R, capital W, but it shows the phenotype red, white flowers. So could you please remember the definition of codominance? The alleles can express themselves independently and equally when they are at heterozygous condition. Here we can found the heterozygous condition. That is why that allele express themselves independently and equally. So the alleles are equally dominant. That is why this cross is called codominance. Then the F1 generations are allowed to self-pollination. So F1 cross with F1, that means capital R, capital W cross with capital R, capital W. Now I am going to develop the F2 generation. Go to the board to develop F2 generation. So capital R, capital W cross with capital R, capital W. The produced gamete would be capital R, capital W cross with capital R, capital W. Then these gametes are going to placed in the Punnett square. Capital R, capital W, capital R, capital W. Then fusion of gamete takes place. Capital R, capital R. The flower color is red. Capital R, capital W. The flower color is red, white. Then capital R, capital W. Then the flower color is red, white. Capital W, the flower color is white. So look at the phenotype of F2 generation. 1 red flower, 2 red white flower plant, then only 1 white flower plant. It reveals that the heterozygote genotype gives rise to the phenotype distinctly differ from either homozygous genotype, So the red white is the phenotype which was given by capital R, capital W, is totally different from the red and white. This is called the codominance. The F1 generation produces F2 progeny in a phenotypic and genotypic ratio 1:2:1. So the phenotypic ratio is 1:2:1. Only 1 red flower plant here, 2 red white flower plant here, only 1 white flower plant. Then the genotypic ratio is again 1:2:1. So the genotype is capital R. For capital R W, the young ones are 2 in number. For capital W, only 1 number. Now I am going to pinpoint the difference between incomplete dominance and codominance. In the incomplete dominance, the heterozygotes alleles are not expressed fully, while in the codominant alleles are expressed independently and equally, and both the alleles are expressed equally and they are stick to their original nature. They stick to their original nature. But in the case of incomplete dominance, they are not able to express the parental phenotype, and they produce the intermediate color pink. So this is the major difference between the incomplete and codominance. Now I am going to explain another codominant experiment with an example called AB blood group. Can we differentiate or identify an individual blood group? In the blood, RBC contains A antigen. The blood group is said to be A blood group. Suppose the RBC contains B antigen, the blood group said to be B blood group. Suppose there is no presence of this both antigen, that blood group is said to be of antigen, and that blood group said to be O blood group. Then the allele for the A blood group is capital I, then the superscript capital A. For writing the allele for blood groups, we have to write like this: capital A, the superscript, capital I, the superscript capital A. For the B blood group, the allele is capital I, the superscript capital B. For the O blood group, capital I, capital O. And these two alleles are dominant alleles. These two alleles are dominant alleles. Capital A and capital B alleles are dominant alleles. Then the allele O is the recessive allele. Recessive allele. Now we are going to discuss the AB blood group only. When both the alleles are present together, that means capital IA, capital IB present together, they are able to produce AB blood group. AB blood group. It is categorized by the presence of A antigen and B antigen present on the RBC. So it is categorized by the presence of A antigen and B antigen on RBC. So that's all about today's video. I hope that video is useful for you. Thank you for watching this video. Thank you.